Sure. Okay, good afternoon. Thank you for inviting me to present in this conference. My name is Mai He. I'm from the Washington University in the United States. So today is my talk. The title of my talk is Checkpoint Inhibitor Markers in Pediatric Tumors with Driver Mutations. Next slide, please. So this slide briefly demonstrates the evolution of cancer treatments. Besides the conventional surgery, chemotherapy, radiation therapy, in the 80s of last century, the target therapy, including the tyrosine kinase inhibitor and the immunotherapy, started to emerge. Next slide, please. So you can see like on the top left is Dr. Stanley Cohen, who is a famous tumor immunologist who proposed the term cytokine during that time, the first wave of immunotherapy. He was one of my PhD mentor. And then in the 2000s, there was a major breakthrough of the target therapy, including EGFR, a TKI for lung cancer. And shown here is Dr. Mark Ladani, who is my mentor during that time in the Memorial Sloan Kettering Cancer Center in New York, USA. At that time, he worked with Dr. Collins doing this molecular testing for the EGFR, KRAS mutations for the lung cancer. And then at that time, when I was a fellow in Memorial Sloan Kettering, Dr. Allison, who was there, he is a major figure for the next wave of the immunotherapy, which is the immune checkpoint inhibitor therapy. For the last decade, he was awarded with a Nobel Prize a few years ago for his major contribution to this checkpoint inhibitor therapy. Next slide, please. So what is immune checkpoint inhibitor or blockade therapy? So briefly to say, tumor cells has this mutation, mainly the somatic and non-synonymous mutations, which could lead to the expression or translation and expression of the neoantigens on the tumor surface. These neoantigens can be recognized by our immune response system as non-self, and then lead to the cytotoxic T cell mediated anti-tumor immune response. Theoretically, if the tumor cells has a defective DNA mismatch repair, there will be more mutation, which can be measured by this tumor mutation burden. Okay, more mutation, more neoantigen, so theoretically will be more cytotoxic immune response. But the binding of some inhibitory receptors and ligands on the cell surface, including the tumor cells, including the immune cells in the tumor microenvironment, these bindings, such as PD1 and PD1, could lead to the inhibition and or suppression of this immune response. The immune checkpoint inhibitor therapy are the molecules such as monoclonal antibodies, which could block this binding and then reactivate this immune response. Next slide, please. So you can imagine, for the key players involved in this process, in this pathway, they could be a marker for this checkpoint inhibitor therapy, so which could include DNA mismatch repair genes and their products, right? The somatic and non-synonymous mutation from the tumor cells and neoantigens load or the expression of these inhibitory receptors or ligands such as PD1, PDL1, and also the main player of the T cell immune response, which represented by the CD8 T cells. Next slide, please. But only this pathway I just described is only a corner of the big picture of the immune response against the tumor cells. But okay, today my talk will focus on these tumor cells markers and the microenvironment. Next slide, please. So as I mentioned earlier in the previous slide, the potential markers for this checkpoint inhibitor therapy could including the PD-L1, PD1, CD8. These could be examined by the immunohistochemistry. All the DNA mismatch repair function status, which could examined by the immunohistochemistry or PCR or the gene mutations by whole exome sequencing. The tumor mutation burden can also be measured by the whole exome sequencing. For neoantigen load, we need to combine the whole exome sequencing and the RNA sequencing. And for the tumor microenvironment, RNA sequencing can provide some inflammatory signature. Next slide, please. So my research interest is in the novel therapy and markers, specifically for pediatric solid tumors. Next slide, please. So there are a few key features for pediatric solid tumors. One is the lack of the predictive biomarkers for response to immunotherapy. This is partially due to the paucity of mutation for these, tumors. So this paucity of mutation, as I mentioned earlier, will lead to kind of low neoantigen and resulting in a low T cell clonal frequency, which is the T cell response. Next slide, please. And this slide demonstrates the tumor mutation burden in pediatric solid tumors compared to the adult tumors. On the left side are the pediatric tumors. You can see, their tumor mutation burden is about 1 to 2 log less compared to the adult tumors. If you can see the different solid tumor types, at the bottom of the purple bar. So my research will focus on some of them, especially those not well studied. Next slide, please. So my research focus on the pediatric solid tumors with driver well-defined or clearly known driver mutations, including the driver fusions. These tumors are hypothesized to have fewer genomic alterations, including the mutations. So my target tumors, including those rare pediatric tumors with a tumor predisposition syndrome, such as the malignant rhabdoid tumor, Synovial Sarcoma, which is kind of like within this malignant rhabdoid syndrome, and the DICER1 cancer predisposition syndrome, which including the pleuropulmonary blastoma, and some even more rare tumors, but they are very high-grade, highly malignant and lacking effective treatment, such as the NUT carcinoma. As a kind of comparison, I'm using the worm's tumor. Worm's tumors genetics are more complicated. Next slide, please. So, specifically for this talk, I have 3 questions. The first question is, what are the expression of these markers in these tumors with driver mutation? So the next question will be, what are the clinical significance of these markers? Since my study was a retrospective study, I cannot directly test, their correlation with the response to checkpoint inhibitor therapy. So I'd like to know what are the clinical significance of these markers correlated to the outcomes, like the survival. And the third question will be the potential mechanism for these markers and the correlation with each other. Next slide, please. So as I mentioned earlier, I will use immunohistochemistry as a major tool to study the PD1, PD-L1, and CD8. At this moment, at the present, the kind of like the most accepted marker is the immunohistochemistry marker for PD-L1 on tumor cells and the PD1 on the immune cells. But from many studies, it shows these IHE star results are not always predictive of the response to the checkpoint inhibitor therapy. Next slide, please. So here shows you the kind of like the materials and methods, the clones for the antibody for the immunohistochemistry, and also for PD-L1. There are specific way of scoring the staining results. The one I put here is so-called the combined positive score, CPS, which is like the PD-L1 positive cells. No matter these are the tumor cells or the immune cells. Okay, so this is combined means it's a combination of all the cells, including tumor cells, including the immune cells, with positive PD-L1 staining compared to the total number of tumor cells. Okay, and there's another way of measuring or evaluating the PD-L1 stains I will mention in the next slide. But I'm also showing you here the way the staining of the PD1, which is cytoplasmic, and the CD8, which is also kind of like cytoplasmic and a membrane. And we are measuring not only the staining within the tumors, but also at the tumor normal tissue interface as demonstrated as the picture in the bottom one. So basically, it's kind of like the black dot is kind of like the interface, but we will measure 5 cells in thickness, to each side. Next slide, please. So this slide demonstrates another way of evaluating the PD-L1 immunohistochemistry, which is called a TPS. So the TPS is expressed by the number of PD-L1 stained tumor cells. So this time it's only tumor cells, no immune cells, divided by the total number of viable tumor cells. Okay, so these are how we perform the immunohistochemistry for this study. Next slide, please. And so this one demonstrated the suggested use. They are cut off in different types of cancers. So you can see some cancers people like to use TPS, especially for lung cancer, and for other 2 types of tumors, people like to use the CPS way, and the cutoff ranging from 1% to 10%. Next slide. So this slide lists the tumors I was studying in current study anyway. So including synovial sarcoma and with the characteristic driver mutation, malignant rhabdoid tumor, pleuropulmonary plastoma, angiomatous fibrous histiocytoma, NUT carcinoma, and the Wilms tumor. Next slide, please. So the first question is to answer whether these tumors with driver mutation are their tumor microenvironment always cold, as mentioned or described by many studies or papers. Okay, so we did the CD8 immunohistochemistry to kind of evaluate, to answer these questions. And from this slide, you can see these different tumors with different numbers, okay, demonstrate probably a range from like a 50% to 100% expression of these CD8 cytotoxic T cells. So our conclusion, obviously, you can see next slide. So even in these pediatric tumors with driver mutations, they're supposed to have less genomic alteration. Their tumor microenvironment is not always cold. Okay, so that's good. And then the next step is to evaluate the expression of other markers, including PD1, PD-L1. Next slide, please. So here I'm showing you some examples. Okay, so this is a malignant rhabdoid tumor, which is a very cellular, very aggressive tumor. As can see on the top, H&E. Next, followed by the PD-L1, which is PD-L1 should be a membranous staining. Okay, and then CD8 and PD1. So here you can see, it's only one example. You can see the primary tumor on the left and metastatic or post-treatment tumor on the right. Seems like the CD8 has more expression in a primary tumor, but this is only one example. And I'd like to tell you, like actually in different tumors, this situation, is different between the primary and the mets. So there are a lot of variation between, different types of tumors. Next slide, please. So this is NUT carcinoma. Again, the PD-L1 demonstrate showing you the membranous staining, and the PD1 here is a red staining instead of brown. And the CD8 also, you can see the tumor has quite many numerous cells with a positive staining for CD8. Next slide, please. So this example, H&E of the pulmonary, pleuropulmonary blastoma. Type 2 means it has a mixed cystic and thyroid component. Next slide, please. And here, with the two powers showing you the membranous staining of the PD-L1 in these tumor cells. Next one, please. Okay, so here we use a double staining of the CD8 PD1 to show you like there are quite a high degree of expression of these markers in these PBB tumors, and the double staining enable us to tell the difference between the immune cells and tumor cells and their expression of these two markers. You can see the PD1 is not always expressed on the CD8 positive immune cells. They are also expressed in some of the tumor cells. Next slide, please. So here is an example of a synovial sarcoma. Again, left side is a PD1, and then PD1 with CD8 again shows you the kind of like expression on both of PD1 on both tumor cells, immune cells. And the right side is the PD-L1. Next slide, please. So for the synovial sarcoma, we had about 21 cases. Here are our results of these different markers, including tumor mutation burden. And you can see most of the tumor mutation burden actually had a range below 5, except one case, which has a very high tumor mutation burden, just like the adult tumor, with a range of 200. And then the rest are the PD-L1, TPS, CPS, CD8, and the immunohistochemistry of the DNA mismatch repair. Next slide, please. So this one actually summarizes these markers in these different tumors. So again, we can see these tumors did have a significant percentage has this T cell immune response accompanied by the expression of PD-L1 and PD-1. Okay. And specifically for the PD-L1 here, you can see if you look at these CPS and TPS 2 columns, you can see they have a range from about 10% to about 70%, okay? So in these tumors, there's a range of PD-L1 expression from 10% to 70%. Okay, next slide, please. So based on current recommendation, if you have a positive PD-L1 staining, although different cutoff in different types of tumors, it is okay to start the checkpoint inhibitor therapy. So at least for my question number one, I can say in pediatric solid tumors, we examined, okay, there is a subpopulation of patients which could have a potential response to the tumor immune checkpoint inhibitor therapy, okay? Next slide, please. And my second question is the outcome correlation. So here I gave you an example of the Wilms tumor. So we studied about 40 plus patients of Wilms tumor, about 60 cases. So on the top left, you can see the different follow-up lengths of these patients. On the lower left, you can see the correlation of PD-L1 expression with the overall survival. While you can see a separation of the survival by these PD-L1 positive versus the negative, statistically, we did not reach a significance. On the right, you can see the PD1 correlation with the certain features, including anaplasia and high-stage disease. Next slide, please. So here we summarize these marker correlation. So these marker correlation provide a certain usefulness for these markers, not in addition to the prediction to the response to the checkpoint inhibitor therapy, but also like the significance with the survival, but also provide potential mechanism to explain the expression. For example, the TMB in some tumors, which are actually very high, although most of the cases are kind of like within the expected range. So why we did a further exploration. Next slide, please. So this one is the DNA mismatch repair genes. Their variants in the malignant rhabdoid tumors. So you can see we definitely have a few cases which harbor the mutations, although functionally not known. At least they do harbor mutations related to these DNA mismatch repair genes. Next slide, please. So within our cohort of the Wilms tumor, we did find one tumor mutation burden up to like about 20 mutations per MEC base pair. So actually this one turned out to be a Lynch syndrome with mutation in the PMS genes. We have another two cases within this cohort with loss of a PMS protein expression by the immunohistochemistry. So you can see we do have, even in the pediatric solid tumor population, we do have defective DNA mismatch in some of the patients. Next slide, please. So our conclusion is like a subgroup of these patients, even with a well-defined driver mutation, supposed to have less genomic noise, they still could benefit from the potential immune checkpoint inhibitors due to either high TMB or positive PD-L1 staining, and a comprehensive panel to kind of cover these markers are recommended. And this, for example, the whole exome sequencing could also demonstrate the DNA mismatch gene variants, which could be different from the immunohistochemistry results. Next slide, please. So back to this, picture again. So to understand better of this, anti-tumor immune response, not only we need to look at these tumor cells, but we also need to look into this tumor microenvironment. Okay, next slide, please. So this tumor microenvironment by inflammatory signature, by RNA sequencing, pathway analysis, and the neoantigen analysis are the work we are right now working on. So these conclude my today's presentation. Thank you for your listening.